Dear colleagues, thank you for the interest to our poster. Here I describe you the peculiarities of our whole pipeline that was implemented in order to reconstruct and to analyze any mutational spectra. Of course, I will talk about all input data, including nuclear genes and mitochondrial genes also. First issue that we need to solve is to construction of homogeneous data sample. It means that the parallel sequences we must disregard, but how it can be done solely on the automatic base. In order to solve this issue, we concentrated our attention on two web resources, the time tree database, this one, which is deposit the topology of species tree that reconstructed mainly on whole genome comparison data, and the ORCAN web resource implemented multiple orthology testing procedures using different bioinformatical methods. The complex many methods based approach for the orthology finding is superior according to the last quest of orthology community tests. That's why we need to incorporate such an computationally intensive step in our pipeline. It is of note that mitochondrial gene analysis does not need the orthology testing due to limited and invariant number of genes encoded in mitochondrial DNA. For the computation time reason, we made the ORCAN orthology testing only for distantly related species. After these tests, we iteratively select out from sample all the probable parallels based on simple reciprocal blast hits. The final step for sample preparation is the compilation of BLAST database of orthologous sequences under our analysis. Many species have no well-annotated reference sequences. So, the second challenge is to analyze data without it. How it can be done naturally? In order to solve this question, we simply used the experience of consensus reconstruction of multiple alignments, by multiple alignments, multiple protein alignments or multiple alignments of nucleotide sequences. The technical tricks is to construct species-specific consensus based on the most probable residue at each alignment positions. The reconstructed species-specific consensus can be used in two ways: for ordering the distances between potential outgroups and in-groups, and to select out potential sequencing errors in in-group, in our in-group under analysis. For example, based on consecutive pairwise comparison of in-group sequences with consensus, it's simple to implement checkpoints. The checkpoints colored by red rectangles here on this slide. Finding sequencing artifacts such as, for example, stop codons inserted in the middle of the protein or in the middle of codon sequences, or multiple sequence-specific missense mutations, or For example, outlier sequence containing maximum number of substitutions. However, the most useful checkpoint for us is the balance checking between the theoretical maximum of mutations in sample and real mutations number. For instance, if these variables are equals or nearly equals, then we will have a problem in mutation spectral reconstruction because this result can be obtained in only one case. This case is the absence of intraspecies phylogenetic relations, or a so-called star phylogeny. In other words, the absence of intraspecies phylogenetic relations talk us about the absence of genealogy and about the presence of random factor of mutation generation. This random factor most probably enrichment by the sequencing artifacts. It's very important for our subsequent analysis. After the identification of biologically meaningful set of sequences, we implemented the common phylogeny reconstruction and ancestral states reconstruction procedures. However, according to novel data, we need not to base only on single best predicted ancestral sequence in each inner node of the tree, but also on the maximal spectra of alternative tree topologies and alternative best ancestral states. In order to do so, we implemented phylogenetic tree reconstruction and ancestral states reconstruction by three alternative approaches: maximum likelihood, the Bayesian phylogenic reconstruction, and the parsimony. Taking the maximal number of alternative and best ancestral sequences are both the best way to count the uncertainty of ancestral sequence reconstruction in connection with protein functionality conservation, and also the best way to take into consideration of all pathways of substitutions, which leads to reconstruction of most accurate final mutation spectrum. The last challenge, but the most interesting for us, is to learn yet undisclosed rules of ancestral state reconstruction that are visible only after mutational spectrum reconstruction. What it means? It means that the optimal model of sequence evolution must include not only the probability of state changes, but also the balance between such changes and the context of substitutions in biologically driven manner. This way, we naturally cope with the over or under optimization of gene and protein evolution model by iterating the optimization of evolutionary models by the analysis of their results. Thank you for your interest in our work, and please ask your questions to the following addresses.